Thank you very much for the opportunity to present at this webinar. My name is Sekar Katharasan, and I'm at Massachusetts General Hospital and Harvard Medical School in Boston. I'm going to be talking to you today about the inherited basis for heart attack and considering how lifestyle might affect one's genetic risk. Let me start by talking about these two men who both had cardiovascular disease. On the left of the screen is a gentleman named James Fix, who lived in the United States, was a very avid runner. He's actually started with, he's credited with starting America's fitness revolution in the 1970s. Unfortunately, Jim Fix died at the age of 52 while jogging in the state of Vermont in the United States. So he died at a very young age of heart attack. On the right is Winston Churchill, who is kind of a poster child for bad health behaviors. He always had a cigar in his mouth. He drank heavily. He was obese. Despite all these bad lifestyle factors, he lived to the age of 90. So these two men, Jim Fix and Winston Churchill, highlight two different concepts. One is gene mutations that might increase risk for disease, and the second is gene mutations that might protect people from disease. And I want to spend the next 30 minutes or so walking you through work that we've done to identify risk mutations as well as protective mutations. Okay, two questions. One is, why do some individuals get disease at an early age, like Fix? And the second is, are there mutations that confer resistance to disease that lead to protection from disease, like potentially in the case of Churchill? Let's start with the risk question. So the problem that we focused on is heart attack, or also called myocardial infarction. It's the leading cause of death worldwide, including in India, and it's diagnosed based on symptoms like chest pain, EKG change, and then blood test called troponin. Based on these three factors, one can diagnose heart attack. If you look in the United States, in terms of risk for heart attack, here on the x-axis is age of heart attack, on the y-axis is the distribution, and it's a bell-shaped distribution. The average age for a first heart attack for a man in the United States is 65, and for a woman it's 72. But some people get heart attack at a very early age, like before the age of 50. The question is, why do some suffer a heart attack at a very early age? And broadly, there are three basic reasons. One is what's called monogenic, which means that a single mutation in one gene is sufficient to cause early heart attack. The second is called polygenic, where the additive effect of multiple DNA variants leads to early heart attack. And the third is lifestyle. And of course, there can be combinations of all three of these. So I'm going to walk you through work that we've done to assess the monogenic contribution to early heart attack, the polygenic contribution, and then the lifestyle contribution. So for the monogenic component, the question is there a gene where a single mutation can drive a patient to early heart attack? The experiment that we did here was we took 5,000 people with heart attack in men before the age of 50, women before the age of 60, 5,000 control individuals who did not suffer a heart attack. And then we performed sequencing across the genome and sequenced every single gene. So this is basically 18,000 genes. And the sequencing is called exome sequencing. This involves sequencing about 30 million bases, and of the 3.2 billion in the human genome sequence. And
continue.
It's about 1% of the
Sorry.
About 1% of the sequence, 1% of the letters, 30 million out of the 3.2 billion represent protein coding genes. So what we're doing here is we're sequencing all the genes, and then we're looking for relationship between mutations in each of these genes and risk for heart attack. Shown here are the results. On the x-axis is what's called the expected p-value distribution. On the y-axis is the observed. On the red line is what's called the null distribution, so that's the chance distribution. You can see that each dot, which represents a gene, each of 18,000 genes, each dot, most of the dots fall along the chance distribution. Some dots, like the one up here at the top, basically are very far away from the null distribution, suggesting that there is association signal in this gene. The top gene in the genome in terms of mutations that confer risk for heart attack is shown here, and that gene is called low density lipoprotein receptor, or LDLR. About 2% of all early heart attack is due to mutations in this gene. About 1 in 200 in the general population carries a mutation, and they are at 4-fold increased risk for having heart attack. And so of the 5,000 people that we sequenced, about 100, it's due to a monogenic mutation, and it's in this gene. If you want to learn more about this work, the reference is given here, and one can read a lot of detail with this paper published in Nature last year. So let's move on to the polygenic model. Here we look at the relationship between polymorphisms across the genome and risk for heart attack. And the study sample size is quite a bit larger. It's about 60,000 cases and 120,000 controls. And we've genotyped polymorphisms across the genome. We're doing genotyping rather than sequencing, and we're now looking for differences in differences in frequency of alleles between cases and controls at millions of polymorphisms across the genome. When you do this analysis, you find there are about 55 specific spots in the DNA sequence that contribute to risk for heart attack. Shown here is what's called a Manhattan plot. On the x-axis is the chromosomes, on the y-axis is the strength of statistical evidence. All of the points above the horizontal line are significant, statistically significant, and there are about 55 specific regions. Each dot here in this analysis is not a gene, but rather a single nucleotide variant. So there are now 55 variants, and you can learn more about this work in this paper published in 2015. There are 55 of them, but each one of them only changes disease risk by a little bit, maybe increasing it by anywhere from 5% to 30%. So modest effect on disease risk, common variants, and there are a lot of them. Given that there are many of them, one can now put together into what's called a polygenic score, a genetic risk score for each individual. In this case, you can make a score by taking the 50 SNPs and asking how many copies of the risk flavor did the person inherit. So a score can range from 0 to 100 for each person. 0 being ideal, having no risk flavors from any of the 50 SNPs, 100 being the worst, having risk alleles at each of the 50 SNPs, 2 risk alleles at each of the 50 SNPs. So if you calculate the score in a very large number of individuals, shown here, this is 24,000 individuals from a study in Malmö, Sweden, you can see the risk score is perfectly normally distributed. So this is a distribution very similar to other quantitative traits in the population, like height or blood pressure. Once you see something like this, then you can ask, what if you happen to be in the one end of the curve compared to the other end? What's the degree of difference in risk? So we bend the population into a top quintile, the top 20% of the score, and call them high risk. Then the bottom quintile, the lowest 20%, call them low risk, and then labeled everybody else as intermediate. Now we're going to look at the relationship of high, intermediate, or low genetic risk to future risk of heart disease in this 24,000 people. They've been followed on average for 14 years. They developed about 2,200 events, and we're looking at the relationship of genetic risk score to future risk of heart attack. What we find is that if you're high genetic risk, your hazard ratio is about 1.85. So 85% increase in relative risk compared to the low genetic risk people. So what this polygenic score analysis tells you is that we can now identify 20% of the population at about twofold increased risk for early heart attack based on the common variant genetic score. So I've talked to you about the monogenic model for early heart attack, the polygenic model, and then lastly I'm going to talk about lifestyle. So we know that lifestyle plays an important role in heart attack risk. 4 factors that do play a role are smoking, body weight, regular physical activity, and diet rich in fruits and vegetables. We created a lifestyle score for each person, and if you didn't smoke, if your weight was less than 30, you had regular physical activity, and you had diet rich in fruits and vegetables, if you had all 4, or just 3 out of the 4, we labeled the group as favorable. If you only had 0 or 1, we called them unfavorable, and if you had 2 of these factors, they were called intermediate. We looked at the relationship between risk of heart attack and the status of lifestyle, favorable, intermediate, or unfavorable. And what I'm showing you here is the relationship between the lifestyle score and future risk of heart attack. On the x-axis is years of follow-up from 0 to 20. On the y-axis is the event rate for heart disease. And what you can see is in red is the unfavorable lifestyle group, in green is the favorable, and the gray is in the middle. And the people in red, so these are people who have 0 or 1 of the lifestyle factors, they have much higher risk for heart disease over the follow-up period compared to those in green who have 3 or 4 of the factors. The difference in risk is about 1.71 between the 2 groups. Okay, so unfavorable lifestyle is associated with increased risk of heart attack. So on the left is the genetic risk, on the right is the lifestyle risk. And the genetic risk, the unfavorable, is about hazard ratio of 1.75. In the lifestyle risk, the hazard ratio is about 1.71. The question that now comes to mind is, how do they relate to each other? And specifically, in individuals at high genetic risk, can a favorable lifestyle counterbalance that genetic risk? Can you offset that genetic risk. We looked at this question, and here's what we found. So shown here on the x-axis is the bins of genetic risk: low genetic risk, intermediate genetic risk, high genetic risk. On the y-axis is the standardized 10-year event rate. And among the people who are high genetic risk, I'm giving you information on their heart attack rates based on their lifestyle score. So an unfavorable lifestyle score and high genetic risk led to a 10.7% heart attack rate over 10 years. However, a favorable lifestyle led to a 5.1% heart attack rate over 10 years. So you can see that within high genetic risk, adherence to a favorable lifestyle cuts the risk by more than 50%. This pattern of lifestyle factors offsetting the genetic risk, this pattern was seen in both the intermediate risk group and the low risk group, suggesting that favorable lifestyle offsets the genetic risk in every genetic risk category, that no matter what your underlying genetic risk, it's important to follow a healthy lifestyle. To summarize then, this portion of the risk section of the talk, I've been able to show you that monogenic mutations in one gene, LDL receptor, confer risk for early MI, that myocardial infarction or heart attack is also polygenic with more than 50 variants identified to date, and a score of these variants can identify people at high risk, high genetic risk. And then lastly, favorable lifestyle can offset inherited risk. Let me move now to the second question of, are there mutations that confer resistance to disease? So let me walk you through the concept here. You take a population of individuals, shown here in black. You look at their genome, and they have average risk for disease. There may be a special group of individuals who carry a mutation, and because they carry the mutation, they're at lower risk for disease. They're protected from disease. So why might you want to know this? It's not because of risk assessment, because they are lower risk for disease. The main reason to actually want to learn about these kind of mutations is the possibility of developing new treatments based on this information. What I mean by that is, if you can develop a medicine that mimics the way the mutation works, then that medicine, if given to a lot of people, could actually lower risk for heart disease or other diseases. So the concept of using these mutations to inspire new medicines development. So this sounds great in practice, but is it actually, are there real examples of protective mutations for a heart attack? Well, there are. The first example came in this gene called PCSK9. About 1 in 50 people in the United States, 1 in 50 African Americans actually in the United States, carries a mutation in this gene. As a result of carrying the mutation, the gene is broken, 50% function, and the individuals who carry the mutation have lower levels of LDL cholesterol, the so-called bad cholesterol, and they also have markedly lower risk for heart attack. So these are special people. They're protected from heart attack risk because of the mutation. These data suggest that if you could develop a medicine that mimics this mutation, then that medicine should work in terms of lowering risk of heart attack. And several pharmaceutical companies were inspired by this observation and set out to develop a medicine that blocked this gene. And 2 such medicines have been developed. These are monoclonal antibodies, injectable medicines that basically block the gene, and they've been proven already to lower LDL cholesterol in people. and they're available for use in the United States right now. There are studies ongoing to test whether the mutations also lower risk for heart attack. Now, beyond the PCSK9, we developed several additional examples of protective mutations, and one of them is in this gene called apolipoprotein C3, or ApoC3. 1 in 150 people carry a mutation in this gene. As a result of carrying the mutation, the gene is broken, so 50% function, and these individuals have lower levels of blood triglycerides, a different fat in the blood. And they also have lower risk of heart attack. They're protected from risk for heart attack. These observations have inspired medicines that seek to mimic the mutation. An antisense oligonucleotide developed by a company is now being tested and has been shown to block ApoC3 in people and also lower the level of blood triglycerides. Time will tell if these medicines also lower risk of heart attack. So an interesting question that follows from this observation is that why are these mutation carriers protected from risk of heart attack? What's the reason for their protection? So it turns out ApoC3 is a gene involved in how you handle the fat that you eat. After a meal, a fatty meal, that fat in the gut gets into the bloodstream and circulates in the bloodstream. The body clears that fat through an enzyme called lipoprotein lipase. ApoC3 puts a brake on that process. It blocks the body's clearance of the fat. And so therefore ApoC3 is a bad actor, and mutations that essentially break ApoC3 seem to lead to protection. So we wanted to test this, and what we did was we took people without the mutation, we took people with the mutation, and gave them a fat challenge. This is dietary fat, about 55 grams of fat, and then measured blood every hour for 6 hours afterwards. Here's what was found. On the x-axis is time, 0 to 6 hours. On the y-axis is a triglyceride. And what you can see is in the people that don't have the mutation, the blood triglycerides goes up from 60 at time 0 to about 160 at time 4 hours. So almost a threefold increase in the blood fat after a fatty meal. So this is what's happening to us with every meal that we eat, unfortunately. Now look what happens in the people who who carry the mutation. In the people who carry the mutation, there's almost no increase in blood triglycerides. The triglycerides barely budges. So these people don't have any functioning ApoC3. The brake on the body's brake on clearing the fat has been removed, and these people clear the blood fat very quickly after a meal, and therefore their blood triglycerides does not go up. Now imagine this happening with every meal that you eat throughout your lifetime, and you can understand why these people are protected from heart attack. Their arteries in the heart are just exposed to much less fat in the circulation than others. And because they're exposed to much less fat, the arteries are exposed to much less fat, there's much less opportunity for that fat to get into the artery wall, build up, cause a blockage, and ultimately lead to heart attack. So now we can see why these people who carry the ApoC3 mutation are protected from risk of heart attack. So beyond ApoC3 and PCSK9, we and others have found at least 3 more genes where mutations protect against heart attack. The genes are shown here at the top. The next are the frequency of the mutations. The next is the blood test that changes. And then lastly is how much heart attack risk is reduced as a result of carrying these mutations. The bottom row shows us that there are companies that are trying to mimic the mutations with medicines, and they're in various stages of development. So overall, I want to summarize by saying the concept of risk. If you have knowledge of your underlying genomic risk, that can be used to deliver existing treatments and counsel toward favorable lifestyle modification. For the protective mutations, they're helpful because they may inspire new treatments, basically new medicines that seek to mimic the mutations and reduce risk of heart disease as a result. Let me close with just a couple of thoughts. One is that a lot of the work, all the work that I've described, has been done in individuals outside of India, almost all of European ancestry. And what we really need to do is focus on doing genomic analysis, understanding the genetic basis of heart attack in Indians. Very little of this study, of this type of study, has been done in Indians in India. And there are projects now underway, like this project called Genome Asia 100K, where there's an attempt to study genomic risk for heart attack and type 2 diabetes in Indians. I wanted to close by thanking members of my research laboratory and collaborators. If you want to learn more about our work, you can go to this website, catharacenlab.org. Thank you.
Yes, sir. Now we will take the questions. First question is from Samrat. What are the major causes of thrombosis?
That's a great question. The question is, what are the major causes of thrombosis? So heart attack occurs based on two phases. One is what's called a chronic phase. Over many, many years, there's a slow progressive buildup of plaque in the artery wall, the arteries that supply blood to the heart, the coronary arteries. And then there's an acute phase where at a given time point, that plaque bursts open. There's a blood clot that forms, this thrombosis that the questioner is asking about, and blood flow stops for more than, if it stops for more than 20 minutes, then the part of the heart that gets blood from that artery, that part of the heart dies, and that's a heart attack. So thrombosis is a major component of heart attack risk. And thrombosis can be studied, and that itself has a genetic component. And the major causes are genetic as well as other factors like injury of the endothelial wall, stasis. So there's a number of other factors as well that lead to blood clotting.
Next question is from Preeti. How do you see the coronary artery disease burden in India?
The coronary artery disease burden in India is tremendous. It is the leading cause of death in India. There are a couple of properties that have been, a couple of observations about coronary artery disease in India. One is it seems to be happening at younger ages than what may be happening in the rest of the world. And very common. The exact reasons why it's so common and potentially having younger ages, the exact reasons are not clear. That the reasons could be genetic, that there's fundamental differences in the genetics of Indians versus others that puts them at higher risk for heart attack. or coronary artery disease. And or it could be lifestyle, that basically over the last 20, 30 years, the environment in which Indians live has changed dramatically from an environment where people were very active and, basically didn't really have enough food or were eating just enough, to now where people are not as active and there's an abundance of food, at least for certain segments of the Indian population, the middle class and the upper middle class. And so those may be the reasons why there's a growing epidemic of coronary artery disease in India. What we really need to do is do research in India to understand the extent to which genetics versus lifestyle is playing a role in the dramatic increase in the rates of heart attack in India.
Next question is from Ashwini. What lab tests are available for genetic assessment of CAD in India?
There are very few lab tests in terms of on the genetic side. So when you think about genetic risk, the best way to assess genetic risk, one of the best ways, is to simply ask about a family history. So if the person has a mother or father or brother or sister who had a heart attack at an early age, so this is men less than 55, women less than 65, so a family history of early heart attack puts that person at increased risk by about twofold. So family history, asking about a family history of heart attack is a good substitute for a genetic test, or can be a substitute for a genetic test. If you wanted to really look further into this monogenic or polygenic model, the monogenic model is typically assessed by sequencing a few genes, and the polygenic model would be done by genotyping 50 or so SNPs. The genotyping test is not available yet in India. The sequencing is available by several companies and may be useful in certain circumstances. Right now, the risk for heart attack is assessed not by genetics, but by looking at what we call traditional risk factors, and that is age, gender, smoking status, cholesterol level, blood pressure. These factors and diabetes status. These factors can be put together into a score, and one's risk for heart attack can be assessed based on that information in any given person.
Next question from Abhay. Does heart failure after first heart attack increase cancer risk?
I'm not familiar with the answer to that question, but I'm not aware of any information that connects heart failure after heart attack with cancer risk. But that's not my area of expertise.
Next question from Keshav. How is cholesterol lowering and blood pressure controlling therapies effect in reducing heart diseases?
That's a fantastic question. These are the two major ways that we have that have been proven to reduce the risk of a heart attack. One, lower the cholesterol. Two, lower the blood pressure. And they seem to work in different ways. The cholesterol lowering directly changes the level of cholesterol in the blood, and therefore much less of the cholesterol is able to get in from the blood into the artery wall to lead to the blockage. The major driver of blockage formation is cholesterol coming from inside the blood vessel into the wall of the artery. Once the cholesterol gets into the wall of the artery, it sets up a chronic inflammatory process that ultimately eventually leads to a severe blockage, or a blockage that breaks open and develops thrombosis and leads to a heart attack. So cholesterol lowering, there's a clear understanding. For blood pressure, the blood pressure, high blood pressure basically injures the lining of the artery, interior lining of the artery, the endothelial lining, and therefore lowering the blood pressure preserves the integrity of the endothelial lining, and that may be the major reason why blood pressure lowering works to reduce risk of a heart attack.
Next question is from Ashwini. How is endothelial lipase related to atherosclerosis? How can we look at the enzyme as a therapeutic target?
That's a very interesting question. Yeah, so endothelial lipase is part of the family of lipases like lipoprotein lipase and hepatic lipase. These are enzymes that basically cleave triacylglycerol from different molecules. And over the years, it's been known that blocking endothelial lipase will increase the level of HDL cholesterol in the blood. And endothelial lipase has been, people have tried to approach it as a therapeutic target to increase HDL cholesterol and hopefully on that basis reduce risk of heart attack. We published a couple of years ago, in 2012, a provocative paper that showed that mutations in the endothelial lipase gene are associated with higher cholesterol, higher HDL cholesterol. But surprisingly, the people who carry these mutations were not at reduced risk for heart attack, were not protected. If HDL cholesterol is a causal factor, then people who carry the endothelial lipase mutations that lead to higher HDL, they should be at lower risk for heart attack. And they were not. They were the same risk as those who didn't carry the mutations. This suggested to us that HDL itself may not be a causal factor for heart disease. And second, that endothelial lipase inhibition may not be a good therapeutic approach to reduce risk of heart attack. Currently, I'm not aware of companies pursuing endothelial lipase as a target.
Second question from Ashwini. How relevant is plaque-2 assay or phospholipase-2 assay for diagnosing the heart attack?
Yeah, those two assays are not appropriate for diagnosing heart attack. Diagnosing of heart attack is mainly done based on the troponin. They have been proposed as a way to measure risk for heart attack, future risk, so as a biomarker of risk. And I would say those two tests are not useful beyond what we do already, beyond measuring the cholesterol, the blood pressure, the weight, the height, the age, the gender, the smoking status, diabetes status. Those are fancy blood tests that are heavily advertised by companies, but I don't think they have a meaningful role in predicting risk for heart attack.
Next question from Prerna. How does hypercholesterolemia affect the heart attack rate?
Hypercholesterol is a very powerful relationship to heart attack rate. So if your cholesterol is in the top 20% of the distribution, your risk for heart attack is about twofold compared to those in the lowest 20% of the LDL cholesterol distribution. And over the years, it's been conclusively shown that lowering LDL cholesterol by any of several different medicines will lower risk of heart attack. It's one of the real key cornerstones of how we reduce heart attack risk to keep the LDL cholesterol low, or medicines.